Good evening, everybody. Welcome back to daily lectures from New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We're on to chapter 12 of the subject of molecular biotechnology, and this deals with vaccines, which is chapter 12. And the subtopics are subunit vaccines that deal with HSV, foot and mouth disease. HSV will be herpes simplex virus. Then we have cholera, SARS, Staphylococcus aureus, and human papillomavirus HPV. And on to peptide vaccines that deal with foot and mouth disease and malaria. Genetic immunization with DNA vaccines, the delivery, and about dental caries, and attenuated vaccines that deal with cholera, salmonella species, leishmania species, herpes simplex virus, and the vector vaccines, that is vaccines detected against virus vaccines detected against viruses directed against bacteria as antigen delivery systems, the summary, and finally the review questions. And now on to the subject matter proper. Well, vaccination protects a recipient from pathogenic agents by establishing an immunological resistance to infection. An injected or oral vaccine induces the host to generate antibodies against the disease-causing organisms. Therefore, during future exposures, the infectious agent is inactivated, neutralized, or killed, its proliferation is prevented, and the disease state is not established. Over 200 years ago, in 1796, Edward Jenner experimentally tested the folklore-based notion that human infection with a mild cattle disease called cowpox would protect infected individuals against the human disease smallpox. Smallpox is an extremely virulent disease with a high death rate. If one survives, permanent disfigurement, mental derangement, and blindness often follow. Jenner inoculated James Phipps, an 8-year-old boy, with exudate from a cowpox pustule. In 2 separate trials after the initial vaccination, the boy was fully protected against human smallpox. This country doctor had discovered the principle of vaccination. Now, communicable diseases such as tuberculosis, smallpox, cholera, typhus, bubonic plague, and poliomyelitis. Poliomyelitis have in the past been a scourge for humankind. With the advent of vaccination, antibiotics, and effective public health measures, these epidemic diseases have, for the most part, been brought under control, as shown in Table 12.1. Occasionally, however, protective measures become ineffective and devastating new outbreaks occur. In 1991, a cholera epidemic struck Peru, producing over the next 3 years approximately 1 million infections and several thousand deaths. Also, for many current human and animal diseases, there are no vaccines. Today, more than 2 billion humans suffer from diseases that theoretically could be curtailed by vaccination. In addition, new diseases for which vaccines might be useful continue to emerge. In recent years, in some developed countries, a small but vocal minority of individuals have refused to have their children vaccinated. These individuals argue that many of the previously common illnesses have been vanquished and that they fear the potential side effects of the vaccination more than the disease itself. In addition, many of these people question modern medicine and instead prefer to rely upon the so-called traditional or natural therapies. In fact, the small number of individuals who are not vaccinated benefit from the fact that the vast majority of other people in society have been immunized, thereby making it difficult for many diseases to spread through a community. However, in communities where vaccination levels decrease below a certain level, there is a real danger of some traditional diseases making a comeback. Modern vaccines typically consist of either a killed, inactivated, or a live, non-virulent, attenuated form of an infectious agent. Traditionally, the infectious agent is grown in culture, purified, and either inactivated or attenuated without, of course, losing the ability to evoke an immune response that is effective against a virulent form of the infectious organism. Notwithstanding the considerable success that has been achieved in creating effective vaccines against diseases such as germ and measles, diphtheria, whooping cough, tetanus, smallpox, and poliomyelitis, there are a number of limitations to the current mode of vaccine production. Not all infectious agents can be grown in culture, so no vaccines have been developed for a number of diseases. Production of animal and human viruses requires animal cell culture, which is expensive. Both the yield and rate of production of animals and human viruses in nature are often quite low, making vaccine production costly. Extensive safety precautions are necessary to ensure that laboratory and production personnel are not exposed to a pathogenic agent. Batches of vaccines may not be killed or may be insufficiently attenuated during the production process, thereby introducing virulent organisms into the vaccine and inadvertently spreading the diseases. Attenuated strains may revert, a possibility that requires continued testing to ensure that the reacquisition of virulence has not occurred. Not all diseases, example acquired immunodeficiency syndrome, AIDS, are preventable through the use of traditional vaccines. Most current vaccines have a limited shelf life and often require refrigeration to maintain potency. This requirement creates storage problems in countries with large unelectrified rural areas. Within the last 2 decades, recombinant DNA technology has provided a means of creating a new generation of vaccines that overcome the drawbacks of traditional vaccines. The availability of gene cloning has enabled research to contemplate various novel strategies for vaccine development. Virulence genes could be deleted from an infectious agent that retains the ability to stimulate an immunological response. In this case, the genetically engineered agent could be used as a live vaccine without concern about reversion to virulence, because it is impossible for a whole gene to be reacquired spontaneously during growth in pure culture. Live non-pathogenic carrier systems that carry discrete antigenic determinants of an unrelated pathogenic agent could be created. In this form, the carrier system facilitates the induction of a strong immunological response directed against the pathogenic agent. For infectious agents that cannot be maintained in culture, the genes for the proteins that have been critical antigenic determinants can be isolated, cloned, and expressed in an alternative host system, such as E. coli or a mammalian cell line. These cloned gene proteins can be formulated into a vaccine. There are some infectious agents that do not damage host cells directly. Instead, the disease conditions results when the host immune system attacks its own infected cells. For these diseases, it may be possible to create a targeted cell-specific killing system. Although not a true vaccine, this type of system attacks only infected cells, thereby removing the source of the adverse immunological response. In these cases, the gene for a fusion protein is constructed. First, one part of this fusion protein binds to an infected cell, then the other part kills the infected cell. Because of less stringent regulatory requirements, the first vaccines that were produced by recombinant DNA techniques were for animal diseases such as foot and mouth disease, rabies, and SCOs, and diarrheal disease of pigs and cattle. In addition, many more animal vaccines are currently being developed. For human diseases, a large number of recombinant vaccines are currently in various stages of development, including clinical trials. Unfortunately, in comparison to the number of new therapeutic agents, very few recombinant DNA-based vaccines have been developed. Why, according to the vaccine producers, does it take so long for vaccines to come to the marketplace? First, While there are 25 major vaccine manufacturers worldwide in 1970, in 2005 there were only 5. Second, vaccines are currently viewed as almost a commodity with little financial incentive to develop new vaccines. In 2005, the worldwide market for preventive vaccines was approximately $8 billion. Third, the US government is a major purchaser of vaccines, forcing discount prices and thereby decreasing the potential profit. Fourth, in 1980, in the United States, good manufacturing practices were introduced into vaccine production, causing manufacturing costs to increase dramatically. Fifth, the transition from conventional to newer processes for vaccine production is expensive and time-consuming, not including clinical trials, so that it is preferable to continue using a more established technology. On the positive side, the focus of the larger vaccine manufacturers on large-scale production has provided smaller biotechnology vaccine companies with a number of niche opportunities to develop and market new products. Finally, since most vaccines are intended to protect large... We'll come to this table later. Let's finish the subject first. Population is a very large amount of money that companies often charge to treat a single individual. Some of the newer therapeutic agents, see Chapter 10, are unrealistic for pricing of a new vaccine. In fact, it is precisely in many poor countries where most individuals cannot afford to pay very much for treatment or immunization, vaccines are needed the most. Subunit vaccines. Vaccines are generally consisting of either killed or attenuated forms of the whole pathogenic agent. The antibodies elicited by these vaccines initiate an immune response to inactive neutralize pathogenic organisms by binding to proteins on the outer surface of the agent. So do vaccines need to contain the whole organism or will specific portions of pathogenic organisms. For disease-causing viruses, it's been shown that purified outer surface viral proteins, either capsid or envelope proteins, are often sufficient for eliciting neutralizing antibodies in the host organism. Vaccines that use components of a pathogenic organism rather than the whole organism are called subunit vaccines. Recombinant DNA technology is very well suited for developing new subunit vaccines. There are advantages and disadvantages to the use of subunit vaccines. On the positive side, using a purified protein as an immunogen ensures that the preparation is stable and safe, is precisely defined chemically, and is free of extraneous proteins and nucleic acids that can initiate undesirable side effects in the host organism. On the negative side, purification of a specific protein can be costly, and in certain instances, an isolated protein may not have the same conformation as it does in situ within the viral capsid or envelope, with the result that its antigenicity is decreased. Obviously, the decision to produce a subunit vaccine depends on an assessment of several biological and economic factors. Herpes simplex virus. The HSV has been implicated as a cancer-causing oncogenic agent in addition to its more common roles in causing sexually transmitted disease, severe eye infections, and encephalitis. So prevention of HSV infection by vaccination with either killed or attenuated viruses. We'll come to this diagram later. Attenuated viruses put the recipient at risk of cancer. Thus, protection against HSV would be best achieved by a subunit vaccine which would not be oncogenic. Right? Well, going back to the subject as shown here, that sentence, let's just read that again. So prevention of HSV infection by vaccination with killed or attenuated viruses may put the recipient at risk for cancer. Thus, protection against HSV would be best achieved by a subunit vaccine which would not be oncogenic. The primary requirement for creating any subunit vaccine is identification of the components of the infectious agent that elicits antibodies that react against the intact form of the infectious agent. The HSV type 1, HSV1 envelope glycoprotein D, GD, is such a component because after injection into mice, it elicits antibodies that neutralize intact HSV. The HSV1 GD gene was isolated and then cloned into a mammalian expression vector and expressed in Chinese hamster ovary CHO cells 4.2, which, unlike the E. coli system, properly glycosylate foreign eukaryotic proteins. The complete sequence of the GD gene encodes a protein that becomes bound to the mammalian host cell. However, a membrane-bound protein is much more difficult to purify than a soluble one. Consequently, the GD gene was modified by removing the nucleotides encoding the C-terminal transmembrane binding domain. The modified gene was then transformed into CHO cells, where the product was glycosylated and secreted into the external medium. In laboratory trials, the modified form of GD was effective against both HSV1 and HSV2. Prevent foot and mouth disease. The primary requirement for creating any subunit vaccine is identification of the components of the infectious agent that elicits antibodies that react against the intact form of the infectious agent. The HSV type 1, HSV1, envelope glycoprotein DGD, is such a component because after injection into mice it elicits antibodies that neutralize intact HSV. The HSV1 GD gene was isolated and then cloned into a mammalian expression vector and expressed in Chinese hamster ovary CHO cells, which, unlike the E. coli system, properly glycosylate foreign eukaryotic proteins. The complete sequence of the ID of the GD gene encodes a protein that becomes bound to the mammalian host cell membrane. However, a membrane-bound protein is much more difficult to purify than a soluble one. Consequently, the GD gene was modified by removing the nucleotides encoding the C-terminal transmembrane binding domain. The modified gene was then transformed into CHO cells, where the product was glycosylated and secreted into the external medium. Laboratory trials, the modified form of GD was effective against both HSV1 and HSV2. On to foot and mouth disease. Foot and mouth disease virus, FMDV, was a devastating cattle and swine and is extremely virulent. But for the most part, it has been possible to keep negative effects of the virus to a minimum by using formalin-killed FMDV preparations as a vaccine. Approximately 1 billion doses of this killed virus vaccine are used worldwide each year. The availability of the vaccine notwithstanding, in 2001 there was a major outbreak of foot and mouth disease in Europe, in which tens of thousands of cattle were slaughtered and the carcasses were incinerated in an effort to prevent the virus from spreading. Research in FMDB found that the major antigenic determinants that induce neutralizing antibodies is capsid virus protein 1, VP1. Although purified VP1 is a much less potent antigen than intact viral particles, it can still elicit neutralizing antibodies by itself and therefore can protect animals from infection by FMDV. Foot-and-mouth disease virus. Thus, the gene for VP1 became a target for cloning. The genome of FMDV is composed of a single-stranded RNA approximately 8,000 nucleotides long. Therefore, for recombinant DNA manipulations, it was necessary to first to synthesize a double-stranded complementary DNA cDNA of the entire genome. This cDNA was then digested with restriction enzymes and the fragments were cloned in an E. coli expression vector. The product of the VP1 coding sequence was identified immunologically as part of a fusion protein under the control of the PL promoter CL repressor system. The fusion protein was 396 amino acids long and consisted of a portion of a stabilizing carrier protein, that is the bacteriophage MS2 replicase protein, as well as the anti-coding sequence of the phage MS2 of the FMDV VP1 protein. The fusion protein containing the VP1 protein fragment was able to generate neutralizing antibodies to FMDV. A fusion protein, however, faces more government regulatory hurdles than intact VP1 because of the potential immunogenic effects of the non-VP1 component. Therefore, the VP1 sequence alone will have to be subcloned into a different expression vector. Nevertheless, a subunit vaccine for foot and mouth disease could soon be ready for clinical trials. Now I think it's already there. If I was there, we'll go for an update on that later. Excuse me. Cholera. The bacterium Vibrio cholerae, the causative agent of cholera, colonizes the small intestine and secretes large amounts of a hexameric enterotoxin, which is the actual pathogenic agent. This protein consists of one subunit, the A subunit, that has ADP ribosylation. Activity and stimulates adenylate cyclase, and 5 identical B subunits that bind specifically to an intestinal mucosal cell receptor. The A subunit has 2 functional domains, the A1 peptide, which contains a toxic activity, and the A2 peptide, which joins the A subunit to the A sub B subunits. Until a few years ago, a traditional cholera vaccine consisting of phenol-killed V. cholerae was in common use. The vaccine generated only moderate protection, typically lasting from about 3 to 6 months. More recently, a vaccine DICORAH consisting of heat inactivated B. cholerae, Inaba classic strain, heat inactivated Agava classic strain, formalin inactivated Inaba L-Tor strain, formalin inactivated Agava classic strain, and recombinant cholera toxin B subunit has come into use. The vaccine is taken orally, 2 doses 1 week apart, and it is claimed that an additional boosted immunization is not required for about 2 years. SARS. In 2003, there were more or less simultaneous outbreaks in several major cities, including Hong Kong, Singapore, and Toronto, of a new unknown disease. The first case of this disease, severe acute respiratory syndrome, or SARS, was reported in Guangdong Province, Southern People's Republic of China, in November 2002. Given the enormous frequency of air travel, the disease rapidly spread to 29 countries on 5 continents. With the assistance of the WHO, authorities in affected regions immediately implemented strict infection control procedures so that by mid-2003, the outbreak was effectively contained. However, this was not before a total of 8,096 SARS cases and 774 associated deaths were reported. Within a very short time, scientists had identified a novel coronavirus as the causative agent of the disease. The SARS virus contains a single-stranded plus-sense RNA genome of approximately 30 kilo base pairs. In nature, the viral spike protein, which is inserted into the viral membrane, binds to a receptor protein that is present on the surface of mammalian host cells, as seen in 12.6 above. You see this? Carbohydrate, receptor, mammalian cell, a receptor, spike protein, viral membrane. Following the binding of the virus to the receptor, the viral and cell membranes confuse, thereby facilitating entry of the virus into the cell. The spike protein, or the external portion of the molecule, is an attractive candidate for the development of a subunit vaccine. In practice, it was found that the external portion of the spike protein, that is amino acid 318 to 510, could bind efficiently to the host cell receptor protein. Following the protein determination of the complete nucleotide sequence of the SARS virus in 2003, it was relatively straightforward to express a codon-optimized version of this 192 amino acid peptide in CHO cells. In addition to encoding the 192 amino acid spike peptide, the DNA construct introduced into the CHO cells also included a mammalian secretion signal and N-terminus Staphylococcus aureus protein A purification tag and a tobacco H virus protein cleavage site. The recombinant protein synthesized in the CHS cell was secreted into the growth medium, purified by affinity chromatography on a column containing immobilized immunoglobulin G, and then digested with the tobacco etch virus protease to remove the protein A purification tag. Using this construct, the spike protein fragment was readily synthesized and purified. To date, the fully glycosylated form of this subunit vaccine candidate has been shown to elicit a strong immune response in mice. It still remains to be seen whether it can protect immunized animals against infection with the SARS virus. Staphylococcus aureus, the gram-positive bacteria. S. aureus is a major cause of hospital-acquired infection. This bacterium produces a pore-forming toxin, is a leading cause of infections out of the bloodstream, lowers respiratory tract and skin, and because of the emergence of antibiotic-resistant strains, is a serious public health threat to address the challenge of treating S. aureus infections. Whole cell attenuated or killed vaccines have been developed. However, these vaccines have not been particularly effective. Similarly, subunit vaccines composed of individual bacterial surface proteins generate immunoresponses that afford only partial protection when tested in experimental animals. However, a more effective subunit vaccine was recently developed to protect individuals against S. aureus by combining several of the bacterium's antigens, starting with the one disease-causing strain of S. aureus. 23 bacterial outer surface proteins were identified to genome DNA sequence data. Then the coding regions of these proteins minus the signal sequences were polymerase chain reaction PCR amplified and cloned into plasmid vectors that enable the process to be expressed in E. coli with the polyhis tag at the N-terminus of the protein to facilitate the production of the overexpressed protein. The proteins were expressed and purified, and mice were separately immunized with each other. Each of the 23 purified proteins, the immunized mice were subsequently challenged by infections of live disease causing S. aureus. Many of the recombinant surface proteins generated an immune response that afforded partial protection against staphylococcal disease, with some proteins affording more protection than others. However, over the long term, immunization with individual spike proteins afforded only modest protection. A mixture of the four proteins that individually generated the most effective antibodies was used to immunize mice and was found to completely protect against the pathogen. The experiment design ensured that only common and not strain specific S. aureus surface proteins were used to immunize mice. Thus, it is not surprising that the tetravalent subunit vaccine that was developed was effective against 5 different clinical isolated strains of S. aureus. This work represents an important first step in the development of an S. aureus vaccine. Human papillomavirus. This is the causative agent of many common sexually transmitted diseases, but most of these infections are benign and often are asymptomatic. Persistent infection with some strains of human papillomavirus is associated with the development of cervical and related cancers as well as genital warts. Since human papillomavirus type 16 is associated with approximately 50% of cervical cancers, a vaccine that prevents human papillomavirus type 16 could significantly reduce the incidence of cervical cancer. Moreover, a vaccine that is directed against several different types of human papillomavirus could effectively prevent nearly all human papillomavirus-induced cervical cancers. To put this cancer among women worldwide. To put this into perspective, cervical cancer is the second most common diagnosed cancer among women worldwide, according to more than 250,000, accounting for more than 250,000 deaths. And now on to peptide vaccines. The question arises as to whether a small discrete domain of a protein can act as an effective subunit vaccine and induce production of neutralizing antibodies. Intuitively, one would expect that only the portions or domains of a protein that are accessible to antibody binding, that is, those on the exterior surface of the virus, will be immunologically important, and that those located in inaccessible regions inside the virus particle could be ignored if they do not contribute to the conformation of the immunogenic domain. If this argument has validity, it is possible that short peptides that mimic epitopes, antigenic determinants, will be immunogenic, could be used as vaccines, peptide vaccine. However, there are certain limitations to using short peptides as vaccines. To be effective, an epitope must consist of a short stretch of contiguous amino acids, which does not always occur naturally. The peptide must be able to assume the same conformation as the epitope in the intact viral particle. A single epitope may not be sufficiently immunogenic. On to foot and mouth disease. Potential epitopes of the soluble antigen FMDV VP1 were identified from the X-ray crystallographic structure and chemically synthesized domains of the protein were tested as candidate peptide vaccines. Peptides corresponding to amino acids 141 to 160 to 151 to 160 and 200 to 213, which are located near the C-terminal end of VP1, and amino acids 9 to 24, 17 to 32, and 25 to 41, which are located near the N-terminal end of VP1, were each bound to a separate inert carrier protein, keyhole limpet, hemocyanin, and injected into guinea pigs. Very small peptides are usually rapidly degraded unless they are bound to the surface of a large carrier molecule. A single inoculation with peptide 141 to 160 elicited sufficient antibody to protect animals against subsequent challenges with FMDV. By contrast, inoculation with complete VP1 or peptide 9 to 25, 17 to 32, and 25 to 41 yielded lower levels of neutralizing antibodies. In an additional experiment, along a peptide consisting of amino acids 141 to 158, joint amino acids 200 to 213 by two proline residues elicited high levels of neutralizing antibodies in guinea pigs. Even when it was injected without any carrier protein, this two-peptide molecule was more effective than either of the single peptides alone and prevented FMDV proliferation in cattle as well as in guinea pigs. Although these results were promising, the dose of peptide material that had to be used to elicit an immunological response was approximately 1,000 times the amount of inactivated FMDV needed to elicit the same response. To overcome this problem, DNA encoding FMDV VP1 peptide 140 to 160 was linked to the gene encoding a highly immunogenic carrier molecule, hepatitis B virus co-antigen HPCAG. When the gene for this fusion routine was expressed in either E. coli or animal cells in culture, the protein molecules self-assembled into stable 27 NM particles of the FMDV VP1 peptide located on the outer surface of the particle. These particles are highly immunogenic in laboratory animals. Therefore, HPCAG may be an effective carrier molecule for such short synthetic peptides. A comparison of the immunogenicities in guinea pigs of a variety of FMDV peptide vaccines, all of which contain the VP1 peptide 142 to 160 sequence, revealed that a fusion protein containing HPC, AGA, and PMDBP1 amino acids 142 to 160 was approximately 1 by 10 as immunogenic as activated FMDB particles, 35 times more immunogenic than a fusion protein containing E. coli, beta-galactosidase, and FMDB BP1 amino acids 137 to 162 and 500 times more immunogenic than the free synthetic peptide composed of amino acids 142 to 160, because synthetic peptides fused to HPC AG do not interfere with the assembly of the 27 nm hepatitis B virus like particles, and because these particles are nearly as immunogenic as the intact virus from which synthetic peptide was derived, this approach may become a general method for the delivery of the peptide vaccines. Malaria. The genus Plasmodium consists of approximately 125 known species of parasitic protozoa, 5 of which are known to infect humans and cause malaria. The Plasmodium life cycle is very complex. Sporozoids from the saliva of a biting female molecule transmitted to either the blood or the lymphatic system and then migrate to the liver and invade liver cells. Parasites. The parasite buds of the hepatocytes in merosomes containing hundreds of thousands of merozoids. These merosomes lodge in pulmonary capillaries and slowly disintegrate there, generally over 2 or 3 days, releasing merozoids. The merozoids invade the red blood cells, where the parasite divides several times to produce new merozoids, which then leave the red blood cells and travel within the bloodstream to invade new red blood cells. The parasite eventually forms gametocytes, which may be ingested by feeding mosquitoes. Fusion of the gametes that develop from gametocytes leads to the formation of new sporozoids in the mosquito that can infect new individuals, spreading the disease. In the life cycle of the malaria parasite, it is the asexual blood stage multiplication that is responsible for most of the acute symptoms of the disease. In areas where malaria is endemic, and some individuals show considerable resistance to the disease, despite the fact that when their blood is examined, they are found to carry the parasite. This resistance to the worst symptoms of malaria was shown to be a result of an antibody-dependent cellular inhibition mechanism that inhibits parasite development. In other words, some individuals who were infected with the malaria parasite made antibodies against the parasite protein that prevented the growth of the parasite. Following a detailed study, it was determined that the protective antibodies targeted merozoite surface protein 3. When this protein was examined in different strains of Plasmodium, it was observed that while the N-terminal part of the protein varied considerably from one strain to another, the C-terminal end of the protein was highly conserved among the various isolates of the parasite. It was therefore decided to chemically synthesize peptides that corresponded to small portions of the C-terminus of merozoite surface protein 3. Human antibodies from individuals who were resistant to the parasite were affinity purified based upon their interaction with one or more of these peptides. The antibodies that bound to the peptides were then tested in an antibody-dependent cellular inhibition assay. Antibodies directed against peptides B, C, and D had a major inhibitory effect on parasite growth based on the ability of peptides B, C, and D to bind to and select protective antibodies. A peptide representing amino acid residues 181 to 276 and merozoite surface protein 3 was chemically synthesized. This peptide is currently being tested in clinical trials as a novel malaria vaccine, but more research needs to be done in the future. Synthetic peptide vaccines could become highly specific, relatively inexpensive, surface safe and effective alternatives to traditional vaccines. Now, the next will be genetic immunization with the use of DNA vaccines. Delivery, a novel strategy that elicits an antibody response without the introduction of an antigen, has been developed. In this case, the gene encoding an antigenic protein is introduced into the cell of a target animal where the antigen is synthesized. In the initial experiments, gold microprojectiles were coated with E. coli plasmid DNA carrying an antigen gene under the transcription control of an animal virus promoter. A ballistic system was used to deliver the microprojectiles into cells in the ears of mice, as shown in chapter 18 for a more detailed description of the polyballistic system. Other workers introduced cloned cDNAs into mouse cells by injecting large amounts of the plasmid carrying the target DNA directly into the muscles of the test animals. However, effective genetic immunization by direct injection into muscles, 100 micrograms per mouse, requires 3 to 4 orders of magnitude more DNA than the biolistic delivery system, 10 to 100 ng per mouse. And one distinctive feature of genetic immunization is that the costly and time-consuming procedure of either purifying an antigen or creating a recombinant vaccine delivery vehicle is bypassed. Moreover, proteins produced by this procedure are more likely to become correctly post-translationally modified than are proteins that are produced by different host organisms. An advantage of genetic immunization, besides bypassing the need for purified protein antigens, that it can trigger a response against only the protein encoded on the plasmid and not against the plasmid itself. And in addition, when plasmid DNA is introduced into a mammalian system, only those genes or cDNAs under the control of eukaryotic regulatory signals will be transcribed and translated. Antibiotic resistance genes for maintaining the plasmid in E. coli will not be transcribed or translated, and the same vector can be used to deliver different proteins to an individual at the same time, or the administration of the same gene can be repeated a number of times. The feasibility of genetic immunization has been examined in detail. In one series of experiments, mice were injected in the quadriceps of both legs with E. coli plasmid carrying the cDNA for influenza A virus nucleoprotein under the transcription control for either a Rose sarcoma virus or a cytomegalovirus promoter. Although the expression of the nucleoprotein was too low to detect, nucleoprotein-specific antibodies were observed in the blood of the test mice 2 weeks after the initial injection. In comparison to control mice, the nucleoprotein-injected mice were significantly protected against the lethal effects of influenza virus infection. Moreover, the nucleoprotein-injected mice were also protected against the direct strain of influenza virus. This cross-protection is in sharp contrast to traditional influenza virus vaccines, which are directed against surface antigens of the virus so that each vaccine is specific to a single strain. Advantages of genetic immunization over conventional vaccines. Table 12.3 shows cultivation of dangerous agents not required. Since genetic immunization does not utilize any viral or bacterial strains, there is no chance that an attenuated strain would revert to virulence. And since no organisms are used, attenuated organisms that may cause disease in young or immunocompromised animals are not a problem. Approach is independent of whether the macroorganism is difficult to grow or attenuate. Production is inexpensive because protein does not need to be produced or purified. Storage is inexpensive because of the stability of DNA. One plasmid could encode several antigens. Vaccines or several plasmids could be mixed together and they are administered at the same time. And now, the effect of that on influenza virus. Let me just repeat that sentence for you. This cross product is in sharp contrast to traditional influenza virus vaccines, which are directed against surface antigens of the virus. So each vaccine is specific to a single strain of influenza virus. In addition, traditional vaccines work only as long as the antigens on the surface of the virus do not change. Unfortunately, the genes for the surface antigens must mutate at a high rate, which creates significant differences among strains. All the core components of the virus, such as the nuclear protein are relatively invariant. They can activate the immune system by a mechanism that is different from that of surface antigens. The fate of introduced DNA is not known and could have the undesirable effect of integrating into the genome of the host cell, possibly disrupting an important gene. However, this risk is currently considered to be extremely low. It is more likely that the DNA will exist for a short period as a non-replicating extra-chromosomal element before it is degraded. To date, genetic immunization has been used primarily to induce immune responses in animals and to a more limited extent in humans against a number of pathogenic organisms, including influenza A virus, human immunodeficiency virus HIV type 1, bovine herpes virus A1, rabies virus, Plasmodium species which cause malaria, hepatitis B virus, hepatitis C virus, bovine rotavirus, bovine respiratory syncytial virus, pseudorabies virus, FMDV, Newcastle disease virus, Clostridium tetani which causes tetanus, and Mycobacterium tuberculosis which causes tuberculosis. Several human clinical trials using DNA vaccines are currently ongoing. One of the problems with the use of DNA vaccines in large animals and humans compared to mice is that transfection efficiency of introduced plasmid DNA is often insufficient to generate a protective immune response. One approach to deliver foreign DNA to animal cells utilizes biodegradable microscopic 0.3 to 1.5 microM polymeric particles with a cationic surface that binds the plasmid DNA. Plasmid DNA is bound to the surfaces of these macroparticles and is slowly released over a period of 2 to 3 weeks after inoculation of an animal. Using macroparticles, it was possible to achieve the same biological effect as with naked DNA with about 250-fold less DNA, demonstrating the potential of this approach. In addition, the level of antibodies induced by the expression of plasmid-encoded genes bound to microparticles was significantly enhanced by 1. Addition of the vaccine adjuvant aluminum phosphate, and 2. The use of nanoparticles 0.05 micrometer in diameter that were coated with poly-L-lysine. In contrast to naked DNA bound to microparticles induced potent cytotoxic T lymphocyte responses at a low dose. To date, most DNA vaccines have been delivered either by intramuscular or intradermal injection. Although these vaccines can induce a potent immune response, they do not induce mucosal immunity. Mucosal immunity can prevent pathogens from entering the body, while systemic immunity deals with pathogens only once they are inside the body. This is an important consideration because mucosal surfaces, including the respiratory, intestinal, and urogenital tracts, are the major sites of transmission of many infectious diseases. However, because of the protective barriers of the mucosal surfaces, traditional antigen-based vaccines are largely ineffective unless they are administered with specific antigens that penetrate or bind to the mucosa, that is mucosal adjuvants. Mucosal immunity induces a separate and distinctive response from systemic immunity. The antibodies produced as part of the mucosal immune response restrict not only mucosal pathogens but also microorganisms that initiate colonized mucosal surfaces and then cause systemic diseases. Many mucosal vaccines are live attenuated organisms that infect mucosal surfaces and are effective at inducing mucosal responses. Of these, old polio vaccines in both attenuated Salmonella enterica, Serovar typhoid Ty 21A, and Vibrio cholera vaccines are licensed for use in humans. DNA vaccines that are designed for delivery to mucosal surfaces are similar in principle to those used for intramuscular or intradermal delivery. To increase plasmid uptake and decrease its subsequent degradation, variation methods of formulating DNA have been tried. For example, cationic positively charged liposomes have been used to deliver DNA, which has a negative charged phosphate backbone to the respiratory tract, and DNA entrapment biodegradable particles have been used for the oral delivery of foreign DNA. Moreover, to improve the potential DNA vaccines for humans, a number of strengths have been devised, including using plasmids that, in addition to encoding a target gene, also expresses a cytokine, such as interleukin-2, IL-2, IL-10, IL-12, which can act as an intracellular mediator, the generation of an immune response. A range of systems including liposomes, live vectors, bacteria and live viruses, and a wide range of adjuvants that increase the immune response. Bacterial toxins, carboxymethylcellulose, lipid derivatives, aluminum salts and saponins have been tried for delivery of DNA to different cell types. Of necessity, various optimization strategies are tested in mice before they are tried on larger animals and then on humans, with no guarantee that an approach that works well in mice will also be a successful strategy in humans. Nevertheless, given the many perceived advantages of genetic immunization over the use of conventional vaccines, this has become a very active area of research. For example, electroporation has been used to increase the transfection of DNA encoding target antigens. With this approach, DNA is injected intramuscularly and the skeletal muscle is immediately electrically stimulated, with a pulse generator with a pulse generator. Despite the fact that this procedure, which causes some patient discomfort, results in a local tissue injury and inflammation, it is tolerated by patients without the need for any anesthesia, and there do not appear to be any long-term side effects to delivering DNA in this way. Most likely, the electrical pulse decreases the transfection efficiency of the added DNA, and it is becoming a method of choice for clinically administering DNA vaccines. A model strain of the invasive bacterium Shigella flexneri has been developed to facilitate the delivery of DNA into animal cells for genetic immunization. Shigella can enter animal epithelial cells, escaping the phagocytic vacuole, and the bacterium can direct plasmid DNA to the nucleus of the host cell, where the introduced genes contain a eukaryotic promoter that is transcribed. Shigella is normally a pathogenic organism and would not be acceptable DNA, to DNA delivery system. Therefore, to use Shigella, it was first necessary to construct a non-pathogenic version of the wild-type organism by engineering the bacterium to be toxin deficient, 2, making a deletion of mutation in the Shigella is the gene which encodes the enzyme aspartate beta semialdehyde dehydrogenase. This enzyme is normally involved in the synthesis of bacterial cell wall constituents diaminopamalic acid. Therefore, the mutant cannot grow unless diaminopamalic acid is added to the growth medium. Shigella strains with the mutation can invade animal epithelial cells and deliver their plasmid DNA. However, once present, the Shigella cells are able to proliferate. Determination of the safety of using Shigella as a vector for the delivery of DNA to animal cells must await the results of human trials. However, the results of experiments with the guinea pigs are promising. The greatest potential advantage of this approach is that the Shigella system DNA for vaccination may be delivered orally, greatly simplifying the delivery of a variety of vaccines. Some limitations of plasmid-based vaccines are: 1, the necessity of strong promoters that function in vivo to selectively transcribe the introduced DNA. 2, lower levels of foreign DNA expression resulting from differences in codon usage between the two introduced genes, often of viral, bacterial, or parasitic origin, and the animal being inoculated. And 3, the presence of antibiotic resistance genetic marker genes on the plasmid vector. To avoid the use of antibiotic resistant markers, researchers have developed a series of minimalistic immunogenetically defined gene expression MIDGE vectors following insertion of the genetic gene of interest into a MIDGE, which is actually minimalistic immunogenetically defined gene expression. And the vector, the antibiotic resistance gene, is excised from the vector and oligonucleotide cap is added. specifically to both ends of the delinearized DNA that contains only a promoter by intron, the gene of interest, and a polyadenylation signal. The remaining portion of this plasmid is degraded by exonucleases. The capped end are resistant exonuclease digestion. This lecture should be over in a minute or so. These were the concluding lines of this lecture. The rest of it will be continued in the next lecture, right? So these vectors have been used successfully as a substitute for plasmid vectors. Vaccination of raises monkeys with DNA encoding simian immunodeficiency virus proteins, followed by a booster with a modified vaccinia virus that encoded many of the same proteins, protected the monkeys against infection by simian immunodeficiency virus. So the DNA that was injected 0 to 8 weeks expresses immune immunodeficiency virus proteins, GAG, POL, WIF, VPX, and VPR, as well as HIV type 1 proteins, N, NV, TAT, and REV. The recombinant vaccinia virus expresses simian immunodeficiency virus proteins, GAG and POL, and HIV protein.